So there's two main mutations that have developed that cause this resistance to occur in tuberculosis. And one class is called target-based mutations, which are basically mutations that occur in the ATPE protein. And they account for about 30% of beta-acquelin resistance. And this is the type of mutation that I've studied in this project. And the other class are the non-target-based mutations. So they're mutations in other proteins. And one particularly important one is the one in the MMP-L5, MMP-S5 efflux pump. And the efflux pump basically is a drug transporter which will kind of send beta-acquelin out of the cell. So that accounts generally for the rest of the resistance. But those can occur in a lot of different proteins, whereas the target-based mutations, they'll occur in just the ATPE one. So as I was talking about earlier, ATPE is actually a gene, but it codes for the subunit C of the ATP synthase. And ATP synthase is a very important protein for almost all organisms. And so it's necessary for proper bacterial functioning in order to generate ATP for energy. So once the ATP synthase is inhibited, then the bacteria will die.
So how does changing one amino acid from aspartic acid to any other amino acid, why does it have such a big influence to the binding affinity?
Yeah, so I looked at the structures of the amino acids. I actually forgot to include in this presentation. Thanks for telling me. The aspartic acid, I think it's a negatively charged protein, an amino acid, whereas all the rest, the alanine, proline, valine, and glycine, all of those that it mutates to are all kind of non-polar side chains. So I would think maybe that could have an impact on how the beta-acquelin is binding there.
But now we have repurposed it to treat MDR and XDR-TB, and in fact, it is now classified as a core second-line anti-TB drug by the World Health Organization, and its treatment success rate for XDR-TB is quite high, up to 87%. Of course, it is associated with several long-term toxicity side effects, such as anemia and peripheral neuropathy. But generally, it is a relatively safe drug and a cheaper one compared to other second or third line treatment. So now, linzolid works by binding to the peptidotransferase center of the 50S ribosomal subunit. So 50S ribosomal subunit is basically the protein-making machinery in bacteria. And how they work is that they bind to the 50S binds to the 30S ribosomal subunit to form the 70S. So yeah, my conclusion is basically I use a largely unutilized database full of whole genome sequencing data, and I was able to find 7 mutations that reduce linzolid binding affinity in silico. They are biologically relevant mutations, and they do not seem to affect protein stability, at least in terms of the 2 large tools that I've used to assess them. Of course, these have to be experimentally validated before we can draw any conclusions with these data. And of course, a major question is whether the mutations I evaluated are real amino acid substitutions or they're simply sequencing error blips in the sea of data that we have. And there's other questions that I have that I didn't have time to explore, such as several common substitutions, such as A167S, that are deemed unstable by MCSM, which I did not include in my analysis, but I think if they are present in a large number in naturally occurring strains, they should not be impacting the growth of the bacteria too much. So I think for future directions, it would be interesting to just explore those outliers and see what their impact on linzolid binding affinity is.
Okay, hi everyone. I'm Michelle, and I will be presenting my research project, Mycobacterium tuberculosis, RPOC, structural basis for rifampicin resistance mechanisms. So first of all, what is tuberculosis? Tuberculosis is an infectious disease caused by Mycobacterium tuberculosis, a species of pathogenic bacteria that is the main causative agent of tuberculosis. Mycobacterium tuberculosis bacteria grow optimally in areas of the body where there is lots of blood and oxygen, which is why it's mainly found in the lungs. However, Mycobacterium tuberculosis bacteria can also spread to other parts of the body, including glands, bones, joints, kidneys, the brain, and reproductive organs. According to the World Health Organization, tuberculosis is one of the leading causes of death worldwide, as shown in this graph, as one of the top 10 global causes of death in 2016, making it a serious global health problem. This is partially because tuberculosis is highly contagious, as Mycobacterium tuberculosis bacteria are easily spread from person to person through droplets spread into the air by coughing or sneezing. Tuberculosis incidence rates are highest in African and Southeast Asian regions, as shown in this map of annual tuberculosis incidence in 2017, represented as the number of new cases per 100,000 population.
And if you will have time, then you should basically proceed it further also, so that it can be changed into some meaningful research, and you should not leave in between. So I think with this experience, even now you can go ahead independently, and also in between you can consult people and further your research in this area. Yeah,
I agree with Dr. Akhtar. I think that was phenomenal, and I can't believe that you didn't have a background, because it didn't come across like that at all. You had a very clear picture of what you were addressing, a very smooth workflow. And I also agree with all the questions Dr. Akhtar asked. So I think that's something to think about. And great job, all of you. I'm spellbound. I don't think at your stage, in like when I was in high school or undergrad, I even thought about such projects. So it's really fascinating that you all are doing this, and I would encourage you to take it further, if not this, but any just similar research projects in an area of your interest. But yeah, definitely explore it, because you all have the aptitude. So
thank you. Yeah, thank you.